Gibson cloning is a variation on restriction digest cloning and restriction digest and ligation cloning that is, in general, preferable because it has better specificity, better yield, better orientation. So let's have a look at how it works. Initially, we're in the same situation as restriction ligation. We have a plasmid, and the plasmid has a multiple cloning site, and we have to linearize the plasmid by adding a restriction enzyme. Let's say it's eCOR1. Either way, we linearize the plasmid. And on one side of the plasmid, I will mark about 25 base pairs as blue. And on the other side of the plasmid, I will mark about 25 base pairs as red. Now, these have no specific features. They'll probably be part of the multiple cloning site. It doesn't actually matter what they are. They're just 25 base pairs. Now, let's look at how we actually design a Gibson insert. So, we design a Gibson insert by making sure that these 25 base pairs are present in the insert. So, for a Gibson insert, I have my 25 BP red, I have my insert, and then I have my 25 BP blue. Now, these are homologous with each other. So you can probably tell there's going to be some sort of recombination going on. But it still won't recombine naturally because these are still double-stranded DNAs. Like so. And it's double-stranded on both plasmids. And because this is outside of any organism, there's no existing recombination proteins to try and make this work. But what the Gibson mix you always has is a 5' restriction 5' endonuclease. And the 5' endonuclease in the reaction mixture will eat away at the 5' side. So on this one, it will eat away at, if I call this 5' and that 3', then the 5' endonuclease will eat away at the top, exposing a long sticky end. And in the same way, it will eat away on the other side, on the 5' end, from the other side. And that also exposes sticky end. And the endonuclease also works on the plasmid. So on the plasmid, I'll be cutting away this part, the top strand there, as opposed to the bottom strand on the insert. And on the other side, I'll be cutting away the bottom strand. Again, I'm cutting away from the 5' end. And it does always work in this particular way. Now, what you can probably see from this is that those are pretty big sticky ends. They can be up to about 25 base pairs, right? So those will religate quite easily. And this is the fundamental idea of Gibson. So there'll be a 5' nucleus and a ligase. And the end product of having that insert and that plasmid is that you get the red's homologous regions recombine, you have your insert, and the blue homologous regions recombine. And one benefit of this, two benefits of this over classical restriction digest is, for one thing, it's orientation specific because these sticky ends aren't palindromes because they were made with completely, effectively arbitrary sequences. And the second interesting consequence is that this is scarless. And again, you might say, oh, well, it's not scarless, it has red and blue. But if you remember where I said the red and blue regions came from, they came from either side of the restriction site. So this red and blue, having concatenated, is just the same sequence as would have been present in the MCF anyway, which is a benefit of this over restriction, digest, and ligation. But this is not where it ends, because Gibson can assemble multiple fragments. As before, the plasmid backbone will have about 25 BP either side that we'll call red and blue. But we can also design multiple inserts. So if I want to design 2 inserts, let's say, So maybe your first one's a promoter, your second was a protein coding sequence. My first insert goes there, and on one side it will have 25 base pairs homologous to this region here. And on the other side, we'll give it a green 25 base pairs homologous. Now, where does that green homologous region come from? Well, it can either come from this insert, so it can just be the last 25 base pairs of this insert, or it can come from the next one, because the next one is number 2. That can be your protein coding sequence, and that will also start with green. So again, this green, in order to make this scarless, should come from the insert. Because it has to be about 25 base pairs long, it also means that Gibson is only appropriate for inserts that are at minimum greater than 30 base pairs, but realistically more than 100 base pairs is preferable. And then on the other side of number 2 insert, we have blue. And you can probably see how this assembles. If I open up my plasmid a bit more, so I can draw it in place. So on this side of the plasmid, which had the red sticky end, red. On the other side, I have blue. And once these have been digested a bit, they'll look like this. Insert 1 will come here and will be inserted. And at the same time, this side will be being restricted and re-added. So then we'll get, on the other side of number 2, we had our blue homologous region. So the blue homologous region is required. Now, this is not an ideal strategy for many fragments because the endonuclease is working away. And if it works for too long, then it starts to encroach upon the insert. And if you don't have your entire insert anymore, then it will assemble wrongly. So, Golden Gate yield decreases exponentially with the amount of inserts that you have, because the more inserts you have, the less chance of them combining in the right orientations in the right ways during the time period that the restriction, that the endonuclease is still eating away only at those 25 or so base pairs that are homologous. So Gibson is better for longer fragments and fewer fragments, and Golden Gate is better for shorter fragments and more fragments.